the next topic.
Well, thank you, sir. Thank you, sir, for this session. Myself, Dr. Shilpi Khanna, and I would like to thank Dr. J.K. Sharma, sir, and Dr. O.P. Sharma, sir, for giving me the opportunity to be the chairperson for this session. We all know that the ecogeological profile of India favors the growth of a number of vectors, and that's where we have these vector-borne diseases, may it be dengue, chikungunya, or malaria, Japanese encephalitis, which are endemic throughout the country. There are certain diseases and certain new entries into our country, for example, the Crimean Congo hemorrhagic fevers and the Zika virus. So we have today with us Dr. Sachin Desai, who is an associate professor in the Department of Community Medicine, Geriatric Medicine also, at Nija Lingappa Medical College, Karnataka. And I can also see that he's holding up some expertise in geriatrics, and he also has many publications to his credit. Welcome, Dr. Desai, and over to you.
Thank you, respected seniors, Dr. J.K. Sharma, sir, Dr. O.P. Sharma, senior faculty from India and all around and from abroad. I would like to thank for this opportunity. Respected sirs and chairpersons, I would just want to begin my presentation on vector-borne tropical diseases, especially the preventive strategies. I begin my presentation with a quote saying that humanity has three great enemies: fever, famine, and war. And out of those, by far, the greatest and the most terrible is fever. This was quoted by Sir William Osler. And the burden of the vector-borne diseases is what we are going to highlight today and how important it is in relation to the various countries that they affect. Malaria, lymphatic filariasis, dengue, leishmaniasis, Japanese encephalitis account for majority of the countries which have population who are at risk of nearly 10 times of those who are infected. And the DALY, that's the disability associated life years in millions, is affected highest for malaria. So there are other diseases as well which are of predominant importance which are of vector-borne etiology. So arboviral infections are those viruses which are maintained in nature through the biological transmission between vertebrate hosts by blood-feeding arthropods. And there are certain salient features which makes them pathogenic and difficult to address. They are having a zoonotic origin where animals are involved with complex life cycles, having non-human primary vertebrate hosts and primary arthropod vectors, which make it difficult to address. So humans and domestic animals are dead-end hosts, which are also an important factor which need to be addressed in controlling the disease. So at the outset, I would want to begin by classifying the arboviruses and sharing certain important features about the 4 or 5 important arboviral diseases, which are of high importance all over the world and their geographical location. And then I would want to highlight about the preventive strategies and the vaccines available. So alpha viruses are the ones which cause chikungunya, flaviviruses cause JE, dengue viruses, AFD, yellow fever, sandfly fever, Rift Valley fever are caused by the Bunya viruses, and Chandipura virus, which was an outbreak in Nagpur because of the bats, is caused by Orbo viruses. So dengue fever is seen in more than 100 countries all over the world except Europe, and there are 50 million cases globally which have occurred even today. There is one case of dengue hemorrhagic fever it's the dengue hemorrhagic fever case which is there in the world which needs hospitalization, and 5 lakh hospitalizations and 30,000 deaths annually, which was reported in WHO health report in 2009. So these are the countries which are being endemic of dengue virus, 10 degrees above and below the equator, which have the Aedes aegypti and the Aedes albopictus as the vectors. Now, chikungunya is also another arboviral disease which is prevalent in the Asian, the Chinese, the Australian, and the African countries, and few parts of Europe. So it was first reported in 1953 in Tanzania, and then there are many other regions which are currently being affected. So it was first in India, reported in Calcutta, then it reappeared 41 years later in Andhra Pradesh in 2006 and in 2007 in Idukki district of Kerala. It's present as an endemic in majority of the states in India. So KFD is one disease which I would be highlighting, which is in the eastern parts of Karnataka, which is in the Shimoga district. It's a disease which is known as monkey's disease, and it had an outbreak in 1983-84 with 69 deaths and 2,167 cases, which is endemic to this region of east part of Karnataka in India. So the vector is the ixodetic tick, and it's predominantly when there is an infected bite by the tick. Yellow fever is the fifth disease with which I'll be explaining about which is seen in the travelers in Africa, in America, and it was first depicted as an epidemic in Kenya in 1992. So it's also endemic in 29 countries, and it's seen in America in the tropical forest between 10 degrees north and 10 degrees south, 40 degrees south of the equator. So these are the regions of the South America and Africa that are prevalent with the yellow fever disease. There are also viral hemorrhagic fevers, which includes the Crimean Congo hemorrhagic fever, Hanta virus, Rift Valley fever, Ebola virus, KFD, yellow fever, and dengue, which have been of an immense importance and need to be addressed because of the fatality and morbidity. AES is one important aspect which was reported in India in 5 states in northern India, where there was acute encephalitis syndrome in age group of 10 years and below. And this was predominantly as an outbreak with 389 cases and 160 deaths with case fatality of 41%. And JE was an important vector in 15% of the cases that were reported. Now, how is it important to address these morbidities and the vector-borne diseases? Addressing them because of their complex nature and the way in which they depict their life cycle, integrated vector control measures are an important aspect through which we can integrate various methods in the form of public health and in the form of preventive measures in the environment, in the tick, and in the persons themselves who are affected. So there are various international organizations with intersectoral coordination between the international agencies, the national agencies, and the various health sectors related to the health, agriculture, environment, and the local government, which would together interact and prevent these morbidities. Now, what are the methods that we would need to adopt to prevent these vector-borne diseases? This is at the public health point of view, and I would also be explaining the vaccines in the upcoming slides. So these are the morbidities, which includes dengue, trypanosomiasis, Chagas disease, Japanese encephalitis, leishmania, lymphatic filariasis, malaria, onchocerciasis, schistosomiasis, and trachoma. And the predominant measures include environmental measures, where we would reduce the source either through the interrupting the life cycle or manipulating their habitat, or removing the proximity of the other vectors and waste management. So, including the disease that includes all of these measures is malaria, predominantly, where all the environmental measures would play an important role. There would also be mechanical measures like improvement in the micro and the macro environment. Then newer methods like interrupting the transmission through polystyrene beads on the water surface and blocking the siphon tubes of the vectors, the respiratory apparatus. There would also be biological methods where we would have larvicides, fungi, and natural enemy conservation, which could prevent the growth of the life cycle. There would also be insecticide, personal protection, and macro environmental protective measures for all of these morbidities, which includes the nets, the personal protective equipment, and repellents. Now, related to the chemical sprays, the efficacy is predominantly for insecticide-related bed nets and indoor residual sprays, which can be adopted in the endemic and the areas which are of high intensity. Now, the prevention and control includes the three measures where we would protect ourselves from the bite, we could protect the adult from being uh, progressing into its life cycle, or we could also interrupt the growth of the larva through various methods like environment, chemical and biological, and then the residual sprays, genetic control, and personal protective equipment. Now, the most important aspect that I would be discussing is the vaccines, which is the latest, which has been depicted as on September 2021, the RTS-AS01, which is the vaccine against the Plasmodium falciparum. So the WHO has recommended widespread use of the RTS-S malaria vaccine, and the ongoing project in Ghana, Kenya, and Malawi has researched over 8 lakh children since 2019, and through various agencies, international agencies, and the WHO and country level partners, there are certain salient features in which the malaria vaccine can be implemented in majority of the countries all over the world. It's feasible to deliver. It helps in reaching the unreached. It has a strong safety profile, which has been seen in 2.3 million doses in Africa in the 3 countries, and it has no negative impact on the uptake of bed nets and other childhood vaccinations as well. So the additional factor is related to the central repeat region of the Plasmodium falciparum, which is against the antigen. Antibodies are developed against the circumsporozoite protein and the T cell epitopes of the circumsporozoite protein, along with the the surface antigen of the hepatitis B virus. So there's a single fusion protein, and which is co-expressed in the E cells with free hepatitis B antigen, and this vaccine can be used with utmost efficacy and efficiency. So the WHO recommends use for the prevention of Plasmodium falciparum in children in those areas with moderate to high transmission, with 4 doses beginning from children with 5 months of age, and then slowly reduction, which will help in reduction of the malaria disease and burden. The exact schedule and the exact route would be described in the upcoming days. Related to the dengue vaccine, a recent news from the NCBS, Tata Institute of Fundamental Research, Bengaluru, where Dr. Arun Shankar Das and Dr. Sudhir Krishna have explained about the study which focused on the envelope domain region 3 of the viral proteins as responses to the segment which were reported to the circumvent antibody dependent enhancement of dengue virus. And this is an important challenge which has been overcome, which in this current vaccine can be used. They've also added the NS1 structural antigen, which is part of the vaccine prevention, and antibodies are developed against this structural protein as well. So the immune profile and response to the novel DNA vaccine has been published, which has been done on a trial in Indo-African countries, and this has been published in the MEDRX4 bulletin, which is on the internet at the moment. So the exact dosage and schedule is yet to be mentioned, but there are other candidate vaccines as well, and the developer, which are there in the rest of the world, which includes the dengue vaccine, the Tetravax, which are live attenuated vaccines. There are also purified inactivated vaccines, which are present, which are in the phase 3 and phase 2 respectively, and phase 1, which is the inactivated vaccine. Now, there are other vaccines as well, which includes the dengue vaccine recombinant protein DNA vaccine, which is using the E. coli and which has been experimented on the mice. Now, as a concluding remark, controlling of these vectors, the most important aspect is integrated approach wherein we have personal protection for where we stop the bite. We have multiple methods of reducing the vector life cycle, which is in the environment, which in the microenvironment, in the macro environment, and we would also want to integrate
Dr. Desai, I would request you to sum up there. We are running short of time. Thank you. Thank you.
Yeah, and so this is my last slide, and I just want to share that integrated vector measures are the best method to address the vector-borne diseases. So thus, addressing the challenges is through intersectoral coordination and proper community empowerment. Thank you.
Okay. Thank you. Thank you, Dr. Desai, for that elaborate information in a very concise form. Thank you. Over to you, Dr. Sharma, sir. I think we can go on with the next session.
Well, I think it was a wonderful session on geriatrics. It's very thoughtful on the part of the scientific committee to give geriatrics its proper place. However, we all 4 speakers are available. If there is any question, we'll be happy to answer. If there are no questions, then we wind up the session and over to Dr. J.K. Sharma for the next session.
Thank you, sir. Thank you.
Thank you very much, sir. Thank you, chairpersons and speaker, for the wonderful session. Now we come to the last session of today's scientific symposium, that is mixed bag. And to invite our chairpersons, I invite Dr. Aram Chabra, Dr. Anil Manchanda, Dr. Girish Verma, Dr. Mukesh Sharma, and Dr. Vinod Malik. And they are all 5 talks, and they are all very, very interesting talks. But my one request to all the speakers and especially to the chairpersons to please stick to the time, because we are already running more than 1 hour late, and Dr. Aram Chabra has given me indication number of times. Now the honor goes on to Dr. Chabra himself. Please invite the first speaker, Dr. Aram Chabra. Just a second, let me see. Otherwise, I'll introduce all. Dr. Aram Chabra is.
Yes, Dr. Sharma. First of all, I would like to thank you, Organized Scientific Committee of the IPF, and especially thanks to Dr. Sharma for giving the opportunity. So today we will start with a very interesting topic, that is the alcohol, drink or not to be drink. We have a very eminent speaker, none other than Dr. Atul Luthra. He is a director and HOD from Fortis Dock Hospital and faculty certification courses of the Diabetes General British BMJ.